Cool. Hi, I'm Jason. I'm from the University of Maryland. I'm a fifth year working with Patro, and today I'll be talking about how we index what we call spectrum-preserving tilings. Thanks to the very great keynote earlier today, we know that sequencing is cheap, abundant, and to many of us, overwhelming to analyze. The cost per human genome has decreased sub Moore's law rate, and if we look at the NCBI RefSeq database, the number of reference sequences has also grown exponentially. And for many of us here, we're trying to answer the question, how can we compactly represent and query these large collections of reference genomes? I'm going to talk about the set of indexes that care about representing reference genomes as k-mer-sets. There's been very, very interesting work from some of us here in the room today about finding small, what we call spectrum-preserving string sets when given an input set of reference sequences. Briefly, an SPSS, a spectrum-preserving string set, is a set of strings U such that every single k-mer in my reference is also in some string in the set U. So I'm going to give you a toy example. So say I have these two references. You will see that every single k-mer in these two references, every single 3-mer, is going to be in one of these two strings, U1 or U2. So here I've shown AAT. The SPSS tells us what k-mers, or which k-mers are in the set of references, but they don't tell us where the k-mers are. And for those of us who care about the locate query, so say for read mapping, alignment, we would like to return all the positions where a k-mer X, here in blue, occurs on a set of references. So for example, if I query this blue k-mer, I care that I return the positions P1, P2, and P3 in the respective references. To connect the dots a little bit, in our paper, we present this idea called spectrum-preserving tiling. And we call strings in an SPSS tiles, and whenever a tile occurs in some reference, our data structure, or our abstraction, the SPT, stores a 3-tuple, a start position, an offset, and a length. Concretely, whenever a tile UI occurs, we say that we want the substring starting at offset W with length L occurs on a reference with position S plus W. This might seem like mumbo-jumbo to you, but I'll show you a picture. So here's a reference that we saw from a previous slide, and we have a set of tiles, or the spectrum-preserving string set. This reference is covered by 3 occurrences of these tiles, the first of which is going to be U2, and we see that U2 has some start position, and 4 bases covers the first 4 bases of R1. And similarly, we can write down these 3 tuples for the other tiles or strings in the SPSS that cover the reference. What's really interesting now is if we define the SPT, we realize that this kind of abstraction immediately implies a very, very modular indexing kind of idea, or a set of modular indexes. And that the locate query that we care about is immediately possible by composing 2 queries. The first is what we call the k-mer-to-tile query. Given a k-mer X, the k-mer-to-tile query identifies where and in which tile a given k-mer occurs. And the second query is the tile-to-occurrence query. We saw how tiles occur in references. The tile-to-occurrence query, given the rth occurrence of a tile I, returns that start, offset, and length 3-tuple. And again, I'll show you a quick picture about how this works. So we have the tiling, and say we want to query the 3-mer CAA. We see that CAA occurs in the tile U2, and for the 0th occurrence, it occurs with the recorded start position plus the offset in the tile, immediately giving us the location of the k-mer. What's really cool is that because of the work that has been done by this community, there are now very efficient data structures for supporting this k-mer-to-tile query that returns the identity of the tile that contains a queried k-mer, as well as its offset. So for example, our collaborator Giulio has this data structure SSHash that require only 8 bits per k-mer for the human genome. It's worth mentioning here that the SPSS was first defined by Professor Medvedev, who's in this room, I think, with us today. There was a really nice talk by Andre earlier at RecomSeq. And in the next talk, we're going to hear about a really cool construction tool for the de Bruijn graph. But what about the tile-to-occurrence query? Are there these nice drop-in algorithms and tools? It turns out that there exist few compression schemes for the tile-to-occurrence query. And if we want to implement this, what we have to do is simply write down every single occurrences of these tiles. So here I show you 4 references covered by tiles. And essentially, for every single position, kind of shown here with a red triangle, I have to write down a position. Whenever I have to write down a position or this 3-tuple, I need to record a log. I need to use a log number of bits. And the number of occurrences here scales at least linearly to the total size of the reference. And the problem is that this table recording positions now dominates the size of indexes. And the reason is there are many more k-mer occurrences than there are unique k-mers. So to complement this kind of modular scheme where we have these nice drop-in algorithms and for one query and we don't have drop-in algorithms for this other query, we came up with this new compression idea for compressing this table utab for specific kind of SPTs based off unitigs. Some of us might know what unitigs are, but I'll give a brief recap. The important details are that the unitigs always occur wholly in a reference. They share k-1 length overlaps, and this means that utab only has to store start positions. Again, here for unitigs, we have to store a log number of bits for every single one of these red triangles. So we try to ask the question, can we store a log number of bits only for some of the unitigs? And I'm here today to tell you that the answer is yes. And graphically, I'm going to show how we do this. Instead of storing the position of every single unitig occurrence, what we're going to do is sample the tiling. And for every single sampled unitig occurrence, here with a black outline, we're going to store a log number of bits. But for every other occurrence that is not sampled, we'll store only 7 bits. So we get this nice result. The retrievals process for where unitigs occur should feel a little bit intuitive. And essentially, to retrieve the position, say here pointed at by a blue arrow, what I'm going to do is I'm going to traverse backwards on my tiling in an implicit manner in our index. You can read more details in our paper, until we reach one of these red triangles, a recorded position. Because we know the length of the unitigs, we know the start position of a sampled unitig, I know the position of my not sampled unitig. What's crucial in getting from log number of bits per unitig to constant number of bits for not sampled unitig is we very carefully store nucleotides for these not sampled unitigs. So again, we have a tiling, and I'm going to zoom in and show you for every single occurrence of a not sampled unitig what we store exactly. For every single occurrence of a not sampled unitig, what we're going to do is we're going to store a predecessor nucleotide for an occurrence of the unitig. So here, UI occurs somewhere in the reference Rn, and I'm going to store the base that immediately precedes it. Similarly, I'm going to store also the successor nucleotide for this occurrence. So here, this successor nucleotide that immediately succeeds the occurrence of UI, the sequence, is A. And we're going to store these nucleotides in these tables that we call ptab and stab, the predecessor and successor tables, such that all the successor nucleotides and all the predecessor nucleotides for unitig UI for all its occurrences are put together. What's crucial here, again, we're going to store here in this toy example, we only need 2 bits to store each of the nucleotides, and that the predecessor nucleotides prepended to the k-1 prefix of a unitig will land me in the previous unitig. So great that we have the storage. How fast is the traversal? Or how correct is the traversal? What's crucial in the traversal is that we're going to do the traversals not on the de Bruijn graph, but on the references itself. And given the rth occurrence of a unitig I care about which unitig precedes me, but also its rank, like which occurrence of the unitig precedes the ith occurrence here, the rth occurrence of UI here. So graphically, we have this. We've stored data about UI and UJ that occur as predecessor-successor pairs occurrences, and I start at the 1th occurrence of UI. I need to know on the reference that I've stored the UI which occurrence of UJ precedes me. We've stored predecessor nucleotides, and we've stored successor nucleotides. If I carefully think about how I've stored these nucleotides, we realize that every single time I've stored an A here for UJ, I see UI, and every single time I've stored a T predecessor for UI, I see UJ. This means that when nucleotides are stored in the order in which they appear in the reference, adjacent unitig pairs, so UI, UJ here, correspond to unique nucleotide pairs, successors and predecessors. And also, these successor-predecessor nucleotide pairs have matching ranks. So here, the 1th occurrence of the pair AT corresponds to the 1th time that UJ and UI also co-occur. So this is somewhere in the reference. What's crucial is that finding out that if I give here the 1th occurrence of UI, finding out that my predecessor with predecessor nucleotide T is UJ is a constant time operation with our hash tables, or the k-mer-to-tile query, is constant time. But also, finding these nucleotides with matching ranks is also constant time via wavelet matrices operating on the stored nucleotides. So now we have a very, very fast atomic traversal step. To ensure that queries are fast, we're going to sample every sth unitig, or approximately every sth unitig. Our scheme randomly samples approximately 1 over s unitig occurrences randomly to bound the expected traversal lengths to a user-given parameter s. And to ensure that traversals backwards terminate, we sample the first unitig in every single reference to guarantee that our correctness. What's interesting is that when we run this on our data, when we index 30,000 bacteria that are representative of the human gut, when we set s equals to 6, we get a very palatable time-space trade-off. The table where we store unitig occurrences is 60% smaller, and when we build an index querying all the k-mers on 100,000 reads, we incur only a 3.66 slowdown. And you can see S now is this knob we can turn when storing unitig occurrences that lets us to trade off speed versus time. And when we set S equals to 3, we get this intermediate performance, as one would expect. So I've spoken a lot about how we have this nice complementary compression scheme for one part of our modular indexes. What if we used the state-of-the-art k-mer-to-unitig query, and for the same data set, compared to the previous generation of index, which is called pufferfish, some of us might be familiar here today, if we combine our new compression scheme for the tile-to-occurrence query with SSH, the state-of-the-art k-mer-to-tile query, the index is less than half the size. And at the time of writing the paper, the index would be $300 a month cheaper to host on AWS. And I'm sure with inflation around the world, the price is more expensive now. So in conclusion, we introduce the spectrum-preserving tiling that immediately implies very modular indexing of k-mer positions on reference sequences. To complement lots of existing work for the k-mer-to-tile query and finding small SPSSs and indexing them, we introduce a sampling scheme for unitigs that requires only 7 bits instead of a log number of bits per not sampled occurrence. And our benchmark indexes is up to 60% smaller, incurring only a 3.66 slowdown, which we argue is palatable. In future work, there are many kind of directions to explore. There's been very, very exciting work recently that have found that finding optimally small spectrum-preserving string sets that contain all k-mers in a reference can be done in linear time. But the question now, if we have to store positions and store an SPT, can we find optimally small SPTs? The second is, now that we have a knob to sample unitig occurrences for time-space trade-offs, can we sample unitigs in a very data-dependent way that allows us for even better time-space trade-offs? So for example, we might want unitigs that land on tax peaks. If we sample them, query to those will be faster. If reads don't land on those unitigs, then maybe we can incur a slower retrieval. And lastly, just for kind of engineers, can we implement even faster unitig traversals? I welcome you to read our paper where we do some tricks to make this constant time operation fast, not only in theory, but also in practice. But the faster we can make that atomic step, the better time speed trade-off we can get, the sparser sampling, the larger sampling parameter we can use. So that's our work. Spectrum-preserving tilings enables sparse and modular indexing. We built the whole thing in Rust. I've been talking to some of you about Rust. So if we want to hear about how you use Rust or want to use Rust, or come chat with me after, you can contact me at this email. The paper is available at this bit.ly link, and you can find our thing on GitHub. So thanks to my collaborators, Giulio, Rob, Jamshed, and the various funding agencies.
Thank you.